Hello and welcome back to the Intro to BioCyc webinar series. I'm Alexander Shearer speaking for SRI's Bioinformatics group. Last time we took a high-level view of things. You can see here we still have the Brucella ovis genome overview sitting here. You'll notice that I've cleverly changed back to E. coli up on the right here, because that's where we're going to be looking. This time we're going to take a look at genes and transcription units, which basically are operons. We're just being technical and nitpicky when we call them transcription units, since a single gene can't be an operon. But you can call them operons. I think it's okay. So let's say we want to talk about threonine biosynthesis, and we want to start by searching genes. Let's go to Search, Genes, Proteins, RNAs, like we talked about earlier. And let's just go for THRA. There we go. And here we are on a BioCyc gene page. So what's in a gene page? Well, at the top, we have the organism, we have the name of the gene. Then we have buttons. The first gives us the nucleotide sequence. Let's take a look at that. Here's the nucleotide sequence. We have the start codon labeled in green, the stop codon labeled in red. We are very clever that way. Let's back up. Following that, we have the Advanced Nucleotide Sequence button, which gives us the option of looking at more or less than the full gene sequence. This can be useful, for example, when you have to do a PCR that brackets a gene. Say you need an extra KB in either direction. Then on the right, we have the Protein Sequence button, that returns the translated protein sequence for a protein coding gene. And there you go. Moving down the page, we have synonyms, which also includes things like ECK numbers and B numbers, and will also include unique identifiers from your genome of interest, probably. We have an ID number that's actually our object ID in the database itself. We have where it is in the genome and a button that will take you to the genome browser centered on that gene. So remember, we saw the browser in the last section. If I click on it now, you'll see that we're in the genome browser and our gene is right in the center, and it's marked with these hash marks to show that it's the gene we followed into the browser, as it were. But let's back up. Continuing on down the page, we have the sequence length in base pairs and then the translated length of the protein, if there is one. We have protein products. We have reactions catalyzed by those products, pathways involving those products, and the genetic regulation schematic. And you also notice we have this green question mark. We have these in a couple places when we think something is a little complicated and we don't want to jam in a giant explanation. Clicking on the question mark will take you through to an explanation in case you've forgotten how something works. In this case, we're looking at the gene regulation schematic, which has our gene, THRA, and the other genes with which it is co-regulated, in this case the other genes in its operon, and it has what regulates them. In this case, we have charged tRNAs, which is an interesting way of saying that these genes are regulated by attenuation based on the availability of isoleucine and threonine. And you can tell that there's downregulation because it's a little stoppy thing instead of an arrow. We'll look at a more complex gene regulation schematic later. Following that, we see Go terms. We actually annotate Go terms to the proteins, but they show up on both protein and gene pages. Each Go term is listed. You can click through to see something about that Go term and other things that share that Go term. There's an evidence code over here. So evidence codes are a way of saying here is the evidence that supports the assertion that this trait or function or role is accurate. And you can see it's showing me the evidence on mouse over. I can also click on it and it brings up a window. And in this case, the window tells me that there's experimental evidence for this Go term assignment. It's from this paper and it's from a purified protein. You can look at the BioCyc site for more of a discussion of how our evidence code works. But let's just close that for now. Continuing down the page, we have multi-fun terms, which is another kind of ontology describing function. We have unification links, which are links out to other databases. So for example, the strain collection, EcoGene, E. coli hub, and expression data. Continuing on down, we have comparison options that I'm not going to address right now, including aligning across multiple genomes, comparing orthologs, and so forth. Down at the bottom, we have the gene in its local context, which is the gene within its operon and showing some of the adjacent genes. And then below that, we have any transcription units, including the gene. In this case, there's one. Some genes are in multiple. So let's click through and take a look at a transcription unit. Here we are on the E. coli transcription unit THR-LABC, which is involved in regulating genes that are involved in threonine biosynthetic processes and so forth. So we can see a bunch of things at a glance. We see the genes that are part of this transcription unit. Up at the right, we see evidence. This is the evidence for this entire transcription unit, and we can always click through on these. We see the transcription start site and the sigma factor it works with, and we see any regulatory relationships. In this case, it's attenuation, and so we see that we have an attenuation point, where a hairpin forms, and we have the impact of having charged tRNAs available, on transcription through this point or not. Below this, we have a summary, and you notice that a lot of these things include publications. So EcoCyc in particular operates on this sort of assertion citation model. MetaCyc does as well, where we'll make an assertion, THR-LABC is regulated in response to the abundance of both isoleucine and threonine, and then we cite the paper that actually showed that. And if I click on this paper, it takes me through, when available, to the PubMed entry for that paper. Neat. Sometimes those PubMed entries aren't available, and in those cases, it'll take you through to a frame we've made for that publication. That is to say, a database object for the publication, since we couldn't link out to one. As we continue down, we'll see a discussion of, it says promoter, it's talking about the transcription start site. There we go. There's evidence for that as well. In this case, it's inferred from a mutant phenotype and mapping of transcription initiation. We continue down to the right and we see a discussion of the attenuation relationship. Let's go back up to the top. And all the citations are collected at the bottom, as you can see. Now this is an attenuation relationship. Let's just take a moment here and look at something else. There we go. This is another transcription unit, and I moved over to it just to show you what it looks like when there's transcription factors involved in regulating a transcription unit. So whereas the attenuation was shown below the line interacting with the stopping point, here we see the transcription factors are shown when it's known actually where they bind on the DNA relative to the genes. Sometimes it's not known and then they're just displayed around where we think the transcription start site is. In this case, they're shown in this sort of red color to say that they're down-regulating the transcription from this operon. And so in this way we can display different kinds of regulatory relationships, including attenuation and transcriptional repression and other forms of transcriptional control. And again, it's important to remind you, you can click through on a lot of stuff in a BioCyc database. Here you can click through on the genes and they'll take you to those gene pages. So now that we've learned how to look at genes and transcription units, it's time to take the next step and think about metabolic pathways. So come back for the next section of this webinar series and follow along with me as we take a look at pathways in the BioCyc database collection.